Hello, this is Joanne Kirchhoff, and I'm presenting a Visual Literacy Pecha Kucha, prepared for the Instructional Design Technology 5035 course, Principles and Applications of Visual Literacy, taught by Dr. Vera Brown. The competency standard from the Association of College Research Libraries that this lesson will focus on is Standard 3. The visually literate student interprets and analyzes the meanings of images and visual media. This lesson's learning outcomes support the next generation science standards performance expectations. They state that the high school students should be able to demonstrate that they can use and gather evidence to support explanations of cell function and reproduction. Have you ever watched a caterpillar turn into a butterfly? If so, you're probably familiar with the idea of a life cycle. The cell cycle can be thought of as the life cycle of a cell. The cell undergoes a series of steps between its birth and eventually creating two new daughter cells. The cell cycle has two primary phases, interphase and mitosis. Interphase is the longest stage of the cell cycle and contains three phases. The G1 phase occurs right after the cell has divided. The S phase is when DNA replication occurs. And in the G2 phase, the cell rapidly grows large enough to move into mitosis. Mitosis is how multicellular organisms like this plant grow. During mitosis, the cell is going to divide into two daughter cells. As you participate in this lesson, pay attention to the different stages of the cell cycle and think about why the cell cycle is important for growth. When you scrape your knee, mitosis replaces the damaged cells with new cells. Skin cells and blood cells are examples of cells that are replaced regularly in your body. As we go through each step of mitosis, consider how cell division can help repair damaged cells. The first step in mitosis is prophase. During prophase, the chromatin inside the nucleus condenses into chromosomes, becoming more visible under a microscope. Each pair of sister chromatids begin to resemble the letter X, and the nuclear membrane breaks down and disappears. The second phase of mitosis is metaphase. In this phase, the pairs of sister chromatids line up across the middle of the cell. Then spindle fibers attach to the centromeres located at the center of each chromatid pair. During anaphase, the centromeres that are holding the sister chromatids together divide, and the individual chromosomes are pulled apart. The individual chromosomes are moved along the spindle fibers towards opposite ends of the cell. Each resulting set of chromosomes contains an exact copy of the cell's original genetic material. Telophase is the last phase of mitosis. In telophase, the condensed chromosomes stretch out to form thin, tangled chromatin. A nuclear membrane forms around each set of chromosomes, and the spindle fibers disappear. Learners need the opportunity to practice new skills and receive feedback on their progress. The lesson will provide real-time feedback to students as they identify and sort the stages of the cell cycle using Nearpod. In this drag and drop activity, learners will sequence the cell cycle phases in the correct order. The sequence will begin immediately after the cell finishes dividing into two cells. Here are the answers to the first drag and drop activity. Notice that the G1 phase occurs directly after the cell division. Also notice that the cell spends the majority of its time in interphase, which includes the G1, S, and G2 phases. In this next drag and drop activity, learners will need to read the descriptions of the cell phases and match them with the corresponding phases and picture of the cell cycle. Some students will use the acronym PMAT to help them remember the steps of mitosis. Mitosis starts after interphase and includes prophase, metaphase, anaphase, and telophase. Are you starting to become more comfortable with the cell cycle? In this drag and drop activity, learners will match the images of cells with the phases of the cell cycle. In order to identify cells in the various phases of the cell cycle, students need to understand what is happening to the chromosomes during the cell cycle. For instance, notice in metaphase that the chromosomes are lining up across the middle of the cell. In this last activity, learners will view this microscopic picture of cells from an onion root tip and identify and label cells that are undergoing interphase and each stage of mitosis: prophase, metaphase, anaphase, and telophase. Notice that most cells in the onion root are in interphase. This is because interphase comprises the largest proportion of a cell's life cycle.